Hello everyone, and once again welcome to the amazing platform of Physics Wallah, where I, Ayushi Agarwal, your botany teacher, is going to teach you the next chapter of class 11th, cell cycle and cell division. Very important chapter, very interesting chapter. But before starting with the chapter, I had a thought in my mind. I would like to share that thought. Maybe this thought is going to change your perception towards life. The thought says, a winner is a dreamer who never gives up. What is this lining means? A winner is a dreamer who never gives up. That means the people who succeed in life, maybe like Mukesh Ambani, Ratan Tata, these all people are successful because they not only dream but they work hard to even achieve those dreams. Simply dreaming, not working hard is not going to become fruitful. So winner are those dreamers, first of all, who never give up and who also put 100% of their efforts to achieve their dream, to achieve their goals. So students, it's a high time, it's a very high time that now all your dreams are going to come true on this very day of your NEET examination. So now if you really want to achieve your dream and if you really want to fulfill your goals, then already dreaming part is done. Now your hard work is going to pay off. So please student, everybody, stay focused, keep working hard till the time you do not achieve your target. And with this simple thought of the day, let's start with today's class, that is cell cycle and cell division. So this part is done. Moving ahead with the introduction part. First of all, the chapter says cell cycle and cell division. Already you have done the chapter cell, the unit of life, and now we are moving ahead with the next chapter, which is just the continuation of the last chapter, that is cell cycle and cell division. In the last chapter, you have studied about what is cell, the structures of cell and the functioning of cell. But now in this chapter you have to study that how the cells increase their number. Already Rudolf Virchow has told you that all the cells, they arise from pre-existing cell by the manner of division. So that line now we are going to study that how a parent cell divides to produce new daughter cell. Now, division of a cell is not a random process. It's not like if any X cell wants to divide, so without preparation, without any activity, it will just get up and divide and produce number of cells. If this is happening, then surely there is some trouble in your body. But in normal condition, there is whole sequence of events taking place in order to let a cell divide. And cell division will only take place whenever and wherever it is required. Clear? And those sequence of events which takes place prior to cell division together are termed as cell cycle. So what is cell cycle? How can you define the cell cycle? The cell cycle means the sequence of all those events like growth of the cell, DNA replication, duplication of cell organelles, and etc. So sequence of all the events which take place during the time of cell division is termed as cell cycle. First point. Cell cycle, cell division is completely under the control of genetics. This whole process is genetically controlled. That means nowhere cell will divide automatically. Whenever and wherever the cell division is required, then only that cell is going to divide. That means it is completely under the control of your genes. Am I correct or not? Okay, very good. So let's read one by one what the statements are written. These statements are directly taken from NCERT. Hence, it becomes very important to read and to understand that what each statement wants to convey. You never know that any of the question can be framed from any of these sentences. So reading NCERT, understanding NCERT, and then solving question is very, very important part. So now let's read. The sequence of events by which a cell duplicates its genome, that means DNA replication. Apart from DNA replication, organelle duplication takes place, energy synthesis takes place, cell grows in size. All these things together is known as cell cycle. Right? Now, next point. These events are under the control of genes. So, whole cell cycle is controlled by your genetic system. Right? Okay. Now, these 2 points I'll discuss while I'll go deep inside the cell cycle. Now, you know what is cell cycle? Cell cycle can broadly be divided into 2 phases. If I want to study the events of cell cycle, then it's a sequential manner, no random. So, cell cycle can broadly be divided first into 2 phases as interphase and M phase. Right? Interphase is actually the long-lived phase of the cell cycle where the cell prepares itself for division. During whole interphase, the cell growth is taking place. Suppose there is a parent cell who is X amount of size. But if that parent cell will enter into interphase, then its size is going to increase. Why? Because the amount of cytoplasm during interphase is increasing. Clear? So during whole interphase, cell grows in size due to various events, and once the cell receives or achieves the complete size, once the cell gets and achieves the full and fledged size, then it escape interphase and enter into M phase where actual division takes place. Correct? So now read the second statement. Although cell growth in terms of cytoplasmic increase is a continuous process, during whole interphase, the cell growth in terms of cytoplasmic increase is taking place. However, there is one phase in the interphase that is only restricted to allow DNA replication. So during complete cell cycle, there is only one stage where the DNA replication takes place. Otherwise, cytoplasmic growth is taking place throughout the interphase. However, DNA synthesis occurs only during one specific stage in the cell cycle. While I'll discuss the details of interphase, I'll tell you its respective substages and where what happens. Then you will notice that actually cell growth is taking place in all the substages of interphase, but specifically if I talk about DNA replication, then it is only taking at one specific stage of the interphase in the cell cycle. Clear? Next, the chromosomes. DNA synthesis is taking place. The amount of DNA during cell cycle is doubled. It is increased. So it is saying that replicated chromosome are then equally distributed to their daughter cells. And for this, for equal distribution of genetic material to the daughter cell, the next phase, that is M phase, is required. That means cell cycle is a sequential event. You can define cell cycle as the sequence of events which are taking place to initiate and to help in cell division. Right? Cell cycle can be broadly divided into 2 phases: interphase, M phase. During interphase, cell prepares itself for division, whereas during M phase, cell actually divides whatever it has prepared inside it during interphase. Now during M phase, it is the time to divide all those material into their daughter cells equally. Right? Now during interphase, whole interphase, Cell growth is taking place in terms of cytoplasmic increase, but the genetic material is only replicating, doubling during one specific stage of the interphase. Right? Now, if I talk about time, then different cells, they have different timing of their cell cycle. Like for example, if I talk about our human cell, then our human cell, the total time it takes to divide is about 24 hours. And like this, if you talk about some other species cell, then their timing will be variable. So you have to remember that human cell divides in every 24 hours. And this time is not fixed for every cell. It is variable, and from species to species it varies. Clear? Okay, very good. So now moving ahead with the phases of cell cycle, let's start with the first, interphase. I'll explain whole interphase and then we'll write about M phase. Interphase is also known as resting phase or preparatory phase. During interphase, actually the cell is not dividing, but it is preparing itself to undergo division. Like for example, if your mother wants to prepare vegetable, like for example palak paneer at your home, so what will she do? Will she go in the kitchen and directly will she start preparing and just come and serve it to you? Or before starting the gas and keeping the vessel on the gas, she'll go to the market, she'll bring paneer, she'll bring palak, and then she's going to wash them, chop them, right? So before just keeping the vessel on the stove, there are so many preparations done behind it, right? So then only your vegetable, your curry can be prepared. So similarly, the cell, if wants to divide, it cannot simply get up and undergo division. Then it will be a wrong division. Daughter cells will not be appropriate. Hence, every cell before dividing has to first prepare itself. And that preparatory phase where the cell is only preparing itself for division is known as interphase. So interphase, also known as preparatory phase. Right? It's a preparatory phase as cell prepares to divide. Now, there is a misnomer that interphase is also known as resting phase. Simply resting is called because division is not taking place. But actually the cell is resting in interphase? No, it is working even more because it is preparing itself, it is duplicating the organelles, replicating the DNA, producing proteins, RNA, nucleotides. So many work is being done by the cell when it is in interphase. But still it is known as resting phase, which you can call that it is a misnomer. Or even if you want to justify that yes, interphase is resting phase, then your answer should be only in terms of lacking division in this phase. Correct? So second point is also known as resting phase, may be a misnomer because cell is not dividing during this stage. Only preparation is being done. Correct? Otherwise, it is a misnomer because cell is not actually resting. It is doing lot of work. Third point. Now, preparation take lot of time, right? Division. See, when your mother has to go to the market, bring spinach, bring paneer, and she has to wash, chop, prepare the curry, and it takes lot of time. But when she serves it to you, you hardly take 5 minutes and you fatafat you eat and then you go and play in the ground. right? So preparation always takes lot of time and actual work takes limited time. So total time of cell division, if it is 24 hours, like for example in human cell, so 95% of those 24 hours will actually be occupied by interphase. M phase only requires 5% of the total time. So that means interphase is the longest one, which consumes about 95% of the total cell division time. Correct? Now, this interphase can be divided into 3 substages, and one by one you have to study all these 3 substages in detail. They are in sequence G1, S and G2. Hamesha always remember these stages in sequence. You cannot keep G2 above than S or S below than G2. No, the sequence has to be followed G1, S and G2. Once G2 is done, then that means interphase is completed. Cell has to escape interphase and enter into M phase. M phase is the actual dividing phase. It is called as the most dramatic phase of the cell cycle because actually here division is going to take place. It takes only 5% of the total cell cycle duration. So first point you will write most dramatic phase. It is the most dramatic phase of the cell cycle. Why? Because actual cell division takes place during M phase. Right? It only takes 5% of the total time. Right? That means if 24 hours are there, then 95% will be occupied in interphase and 5% will only be occupied during M phase. Now, this M phase can be further divided into 2 steps. M phase can further be divided into 2 steps, which is first nuclear division called as karyokinesis and then cytoplasmic division called as cytokinesis. Correct? Yes or no? So, this M phase can also be divided into 2 broad categories as karyokinesis, where nuclear division takes place, right? And then followed by cytokinesis, where the cytoplasm undergoes division, finally dividing the parent cell into its respective daughter cells. This karyokinesis is again divided into 4 stages in sequence. They are prophase, metaphase, anaphase and telophase. So overall, how many stages are there in cell cycle? Starting from G1, then S, G2. Once G2 is completed, cell will enter into M phase. M phase means prophase, metaphase, anaphase, telophase, and then finally division of the cytoplasm, completing the cell cycle. So if you want to represent this cell cycle in the form of a diagram, then how will you depict it? Very important. 2 years back, question on this diagram was asked. This diagram I have taken from NCERT, which I am going to draw it in front of you. Please pay attention, and yes, it is very, very important. All the substages one by one we will discuss in detail. So for example, you are having one cell, parent cell. It is going to undergo cell cycle and thus it will divide. Finally, once it completes the cell cycle, it divides into daughter cells, right? Now, if this cell wants to enter into cell cycle, it is first going to join the G1 phase. After completing the G1 phase, it is then going to enter into S phase. Once completing the S phase, it will enter into G2 phase. So you can see that major portion of the cell cycle is actually being completed in interphase, which is approximately 95% of the total time. Correct? Now remaining 5% of the time is completed in M phase, which is divided into two steps, that is karyokinesis followed by cytokinesis. So this is your cytokinesis. Karyokinesis can be subdivided into four stages: prophase, metaphase, anaphase and telo. Pro, meta, ana and telo. Correct? And once cytokinesis is completed, the cell ultimately divides into two daughter cells. So this is how the cell cycle. So this is your M phase. Yes or no? Clear to everybody? Which takes only 5% of the total time. So this is how the cell cycle has to be depicted. Now my dear students, is the cell always dividing? No. Is my skin cells right now dividing? Maybe no. If not, then where are they present? So such cells who do not want to proliferate, who do not want to divide, escape the cell cycle through G1 phase and enters into a sort of resting phase called, or quiescent stage called G0. Now some cells, they remain in G0 permanently, but some cells can also again come back to the cell cycle as per the requirement through G1 stage. So the cells who wants to divide keep on rotating in the cell cycle. The cells who do not want to proliferate further, rather they want to perform function, they escape the cell cycle through G1 phase and enter into a quiescent stage called G0. Here the cells can rest temporarily or even permanently. So this is how the cell cycle seems to be like in a diagram form. Now we are going to study all these stages one by one, starting from G1. So substages of interphase, first is G1. G stands for growth or gap. So this is growth 1, or also you can say gap 1 phase. This is the first phase of interphase where actually cell growth is taking place in terms of cytoplasmic increase. Why? Because there is a synthesis of proteins, RNA, nucleotides. Organelle duplication also takes place during G1 phase. So G1 phase is characterized by cell growth. Cell growth in terms of cytoplasmic increase. Right? How? Because during this phase, certain organelles like endoplasmic reticulum is undergoing duplication. So here you can write organelle duplication takes place. Apart from this, like endoplasmic reticulum. Apart from this, then there is also synthesis of proteins required for cell division, nucleotides, like in the form of energy, ATP, GTP, and etc. So thus, G1 phase is characterized as simply a growth phase. But during G1, there is no DNA replication. For DNA replication, the next very important stage is S phase. S stands for synthesis phase. S stands for, my dear students, synthesis. Now what happens during S phase? During S phase, in the cytoplasm, there is duplication of centrosome cell organelle as well as synthesis of specific protein called histone. Other proteins are also getting increased, but the specific one which I am mentioning over here is histone protein. So you have to remember that term. So during S phase in the cytoplasm of the cell, what is happening? Organelle duplication. Which organelle? Centrosome. Centriole is getting duplicated. Apart from this, synthesis of proteins takes place. Right? Which protein I should mention specifically? Histone proteins. Correct? Now, coming to the nucleus. Here what happens, very important students, if you understand this little bit small concept now, it is going to become very easy once we start with mitosis and meiosis. See, once the cell was in G1 phase, the genetic material, the chromosomes are actually present in the form of thin thread-like chromatin fibers. You remember cell chapter? I told you that during interphase, the genetic material is represented by thin thread-like chromatin fibers. Right? Suppose I am talking about a diploid cell. So suppose this is a diploid cell. Right? A diploid cell means having two complete sets of chromosome. Like if I talk about my skin cell, which is a diploid cell, it is having two complete sets. One complete set have 23 chromosome, the other complete set have another 23, and total it becomes 46 chromosome. Okay, so I was talking about a diploid cell. Suppose a diploid cell I have, for example, as I told you, my skin cell that is having two complete sets of chromosome. Now the two complete sets of chromosome, I am going to represent it like this. See, suppose this is one complete set and this is another complete set. Both the sets, the genetic material is present in the form of thin thread-like chromatin fiber. There is upper arm, lower arm attached with centromere, but those arms are thin thread-like. Now, what happens during G1 phase? These diploid cell, these two complete sets remains as it is without any change in them. But once the cell escapes G1 phase and enters into S phase, what will happen to the genetic material? DNA replication will take place. The amount of DNA is going to double without changing the number of chromosome. Right? The number of chromosome will remain 46. But however, if initially the DNA content is 2C, after S phase it will become 4C. The number of chromosome will remain 46, but suppose initially the amount of DNA was 20 picogram. So after S phase it will become 40 picogram. See how? A diploid cell. Now DNA replication will take place. Each chromatin fiber is going to produce one more copy of itself attached to the same centromere. Right? Now it is going to become like this. Initially one complete set was represented by single thread from each centromere, but after DNA replication one more thread will be attached complementary to the existing one. So number remains same, one complete set, another complete set. So still the number is 2n only, 46. But now the amount of DNA has doubled. Initially it was 2C, so here it becomes 4C. Clear to everybody that what happens during S phase? Cell growth is taking place. S phase is also characterized by cell growth in terms of cytoplasmic increase as well as nuclear increase. Cytoplasmic increase, synthesis of protein and duplication. Nucleus में DNA replication but without changing the number of chromosome. Now moving ahead, after S the cell is going to enter into G2 phase. what will happen? G2 phase is also just like G1 phase, where cell growth is taking place only in terms of cytoplasmic increase by allowing protein synthesis, specifically tubulin protein is synthesized during G2 phase, along with organelle duplication like mitochondria and chloroplast. They duplicate during G2 phase and then allowing the cell growth, but without changing the number of chromosome or amount of DNA. So G2 will also be represented by diploid cell and amount of DNA as 4C, because already replication has been done in S phase. So once the cell escapes S phase, enter into G2 phase, the amount of DNA as compared to G1 has become double, but the number of chromosome remains the same. Clear? Yes or no? Very good. So what is G2 phase? G2 phase means, first of all, it is characterized by cell growth. Cell growth takes place in terms of cytoplasmic increase. How? Letting the synthesis of proteins, specifically tubulin protein, is synthesized during G2 phase. Then organelle duplication, which could not duplicate in G1 and S like mitochondria, will duplicate in G2. So organelle duplication takes place, like for example mitochondria, which could not duplicate in G1 phase, will duplicate in G2 phase, and the amount of DNA as compared to, in comparison to G1 is double, is twice. That means if G1 is having 2C, then it will have 4C. Clear? So this is the amount of DNA which was present in G1 and this is the amount of present in G2. Why? Because in mid there was S phase. I hope the thing is clear. However, the number of chromosome will remain the same. So this is about your interphase. Interphase is done. Now moving ahead, there is one more stage but not the part of cell cycle, that is G0 or quiescent. I already have shown you in the diagram. So come back to the diagram once. So G0 is not the part of cell cycle. When the cell wants to suspend its division or does not want to further divide or want to exit the cell cycle, then it enters into quiescent stage or G0 stage. So talking about G0 phase first and then we will come to M phase. G0 phase means also known as quiescent stage. Right? Now when the cell does not want to divide, does not want to proliferate. So when cell do not want to divide, when the cell do not want to divide, escapes the cell cycle, right? Through which phase? Through G1 stage. Through G1 phase and enters into G0 phase. During G0 phase, the cells are not dividing. They are sort of permanent cells which have lost their capacity of division, but now they are rather performing some specific function. Now there are two types of permanent cells. You already know, you are students of anatomy, that some cells who have temporarily lost their capacity of division, like for example my skin cells, and some cells who have permanently lost their capacity of division. Those who have permanently lost their capacity of division, my dear student, they remain in G0 phase once they enter into it. Right? They cannot re-enter into the cell cycle. But The cells who have temporarily lost their capacity of division as and when required, they can re-enter the cell cycle through G1. Divide, complete the job, again exit the cell cycle, suspend the cell cycle and enter into G0 phase. Like your skin cell. Suppose right now my skin is perfect, but if some damage occurs, then healing is required now, and for that new cells are required. Then my existing skin cells who are currently in G0 phase will enter into G1, produce new cells, heal my injury and again come back to G0. Right? Yes or no? Very good. And one more thing, my dear student, G0 phase is also referred to as resting phase sometimes. But this is also a misnomer, because during G0 phase also, cell may not divide, but still it is metabolically active because it is performing function. So here you will write cells are even metabolically active during G0 phase. So calling G0 also a resting phase is misnomer like that of interphase. I hope till here interphase G1S, G2 and what is G0 is clear in everybody's mind. And if yes, then let's start with M phase. Clear? Now, M phase, as I have already told you, it is the most dramatic period of the cell cycle that only requires 5% of the total time of cell division. Most dramatic means preparation is already being done. Now the main job of division, main job of dividing parent cell into new daughter cell is going to happen during M phase. Correct? So it is the most dramatic phase where you can see all the drama happening, condensation of genetic material and so and so. We'll teach you one by one everything. Clear? Very good. Now this M phase, as I have already told you, can be divided into two broad stages. Karyokinesis, where nuclear division takes place. Cytokinesis, where cytoplasmic division takes place. Now, first talking about karyokinesis. There can be two ways of nuclear division, and sometimes we also say there can be two types of dividing phases. To be more specific, there are two types of nuclear division or karyokinesis, that is mitosis and meiosis. Correct? So there are two ways how nuclear division can take place. Once nuclear division has taken place, ultimately cytokinesis comes, which is same in both the cases. So first we are going to study about mitosis type of karyokinesis, and then we will study meiosis type of karyokinesis. And from here onwards, the importance of the chapter becomes even more better. So why to waste time, and let's directly jump into mitosis. Oh, I have given heading as meiosis. Please just change this and make it as mitosis. My error, human errors. Humans are full of errors, and it's always good that when you rectify your error, you accept your mistake and you have courage to get them even corrected, right? So never be shameful of your mistakes. Accept it with full heart and try to rectify it and try not to repeat them. So like here I wrote meiosis, but actually I was about to start mitosis, right? Okay, so what is mitosis? Mitosis is a type of karyokinesis where equational cell division takes place. Now, what is equational cell division? Why are we using the term equational for mitosis? So before I explain you these things, let me tell you one more point. Mitosis can take place in both haploid as well as diploid cell. A cell who's having one complete set of chromosome can also show mitosis, and surely a diploid cell, a cell which is having two complete sets of chromosome, can also show mitosis. right? Chalo, so before I start with the main process of mitosis, first I will explain you that why it is known as equational cell division. So if I am having a parent cell, I think white color will be better. Okay, so now first I am having a diploid cell which is having how many complete sets of chromosome? 2. This is a cell which is in G1 phase, right? An amount of DNA is also supposed to see. Now, after G1 phase, the cell will enter into S phase, where DNA duplication takes place. So, amount of DNA becomes 4C, but since the number of chromosome does not change, so the ploidy remains 2N, sorry. Right, now it is going to enter into G2 phase. Again, same 4C, 2N, same as S phase. Now, my dear student, after completing G2 phase, this cell is going to enter into M phase. Suppose mitosis type of karyokinesis is taking place, then in that case, this parent cell will divide and produce 2 new daughter cells. So during the time of mitosis type of karyokinesis, at the end, one parent cell produces 2 daughter cells which are genetically identical to each other. How? Because both the daughter cells receives the same amount of genetic material as present in their parent cell. Correct? Yes or no? Okay, so what happens after mitosis? Both the daughter cells will become 2N, 2C. 2N, 2C. They will receive 2 complete sets of chromosome. They will receive 2 complete sets of chromosome having 2C amount of DNA. So your initially parent was also 2 and 2C, and finally both the daughters which you achieved, they also have 2C amount of DNA with diploid condition. So you can see that the parent and the daughters are identical to each other. That is why this division is known as equational, as both the daughters are receiving equal amount of genetic material. So you can say that in case of mitosis, why it is equational division? Because both the daughters at the end of division receives equal amount of DNA. As that of parent. Clear? Yes or no? So that is why this is known as equational cell division. Till here you have studied. Now you have to study that how this event is taking place. So now let's start with mitosis in detail. You know, mitosis is a type of karyokinesis which is divided into 4 substages: prophase, metaphase, anaphase and telophase. So first we are going to study prophase. My dear students, prophase is the stage which directly comes after the cell completing G2 phase. So imagine if the cell has just entered prophase, you can also called as early prophase. So how will it look like if you're observing it under the microscope? If the cell is present in early prophase, it has just entered the prophase completing G2, then if you're looking it under the microscope, how will it appear to be? See. So what will happen? There will be suppose I'm talking about an animal cell. So there will be a plasma membrane well-defined genetic material called nucleus. Apart from the nucleus, cytoplasm will be there. In the cytoplasm, all the cell organelles will be found, but in the double amount. Suppose initially there was 1 ER. So in prophase, 2 ER. If it was suppose 1 centriole, then 2 centriole. If there's 1 Golgi, then 2. All the organelles will seem to be doubled, and the genetic material will also be the amount. The number will remain same, but the amount of genetic material will also get doubled. But the genetic material will appear to be like all the threads will be intermingled, and you cannot just identify. Can you see how many fibers, how many chromatin fibers have I drawn in this circle in the nucleus? No. So now we are talking about prophase, which is the first step of karyokinesis. That is the first step of mitosis. Now, as I was telling you, if the cell has just entered the prophase, then you can see how the cell would appear like under microscope. It will have genetic material, double the amount of genetic material, but all those genetic material will be present in entangled manner. There will be no clear distinction that how many fibers, how many chromatin fibers are present in this. So this is how the early prophase cell will look like. In the cytoplasm, all the cell organelles would have doubled, and the genetic material in the nucleus would have also doubled in terms of DNA replication, but not in terms of number. But nothing is as such visible because everything is entangled. Now, as prophase cells start, so it starts with certain events. One of the major event of prophase is condensation of the genetic material. The genetic material which is present in the form of thin thread-like, it start getting condensed to form thick rod-like structures. Correct? So one of the major step that takes place during prophase is condensation of genetic material. Apart from condensation of the genetic material, the nuclear membrane which you can see, the nuclear membrane which you can see along with other duplicated cell organelles, they also start getting disintegrated. The cell organelles along with the nuclear membrane will get disintegrated. Second event. Third event, centrosome which has duplicated its number now will start moving towards the opposite pole. Third event. Fourth event. Now, the microtubules which are present in the cytoplasm, they will join together to form thread-like structures called spindle fibers. So, these are certain events that takes place during prophase. Now, let's try to read them from here. Number 1, prophase actually is the first stage of karyokinesis that follows the G2 phase of the interphase after S phase. You know this thing. Second, it is marked by the beginning of the, as I told you, condensation of the genetic material. Right now, genetic material are present in thin thread-like manner. Now, both the chromatin fibers of the centromere will undergo condensation to form chromatids. And 2 chromatids on one centromere forms your chromosome. And these 2 chromatids of the same chromosome are known as sister chromatid. Correct? So condensation of both the threads will take place to form full-fledged chromosome. So this event takes place during prophase. Apart from this, you can see that centrosome, that is the cell organelle, which is already duplicated during the S phase, it also start moving towards the opposite pole, along with the cell organelles as well as the nuclear membrane disintegrate during the end of prophase, as well as spindle fiber formation also takes place. So if I have to draw the complete cell once it has completed the prophase, then see how my cell will look like. This is the condition of the cell when it has just entered the prophase. Now I am going to show you the cell which is present in its late prophase, or you can say towards the beginning of the next stage, that is metaphase. So during late prophase, first of all, centrosomes, the cell organelles which duplicated during the S phase, will occupy the pole region. Right? These centrosomes, once they reach towards the opposite pole, they give rise some microtubular asters. Proper star-like appearance. So they produce microtubular asters, and these asters helps in the attachment of spindle fibers, which are also made up of microtubules. So asters as well as spindle fibers, together they make mitotic apparatus. Clear? Nuclear membrane and your centrosome and other cell organelles will get disintegrated. They will not appear toward the late prophase or towards the beginning of early metaphase. And the genetic material which is now present in the form of chromosome will hang themselves to these spindle fibers with the help of their kinetochores. So with the help of the kinetochores, the chromosomes will hang themselves, all 46 chromosomes will hang themselves to the spindle fibers. Clear? So this is how the actual cell will look like once the late prophase or early metaphase is going to begin. So what are the events? Movement of centrosome towards the opposite pole, formation of aster that lets the attachment of spindle fiber together called mitotic apparatus, condensation of genetic material. Now my dear students, once the condensation is done of the genetic material, then actually you can count the total number of chromosome, which was initially not possible, as genetic material was not having its specific identity. They were all entangled, they were forming a mass of threads. But now once the condensation is done, then actually you can count the total number of chromosomes present in a diploid or if it is a haploid cell. But here I am taking an example of diploid cell. Clear? Cell organelles, they get disintegrated. Nuclear membrane, nucleolus also disappears. Now this is the events that takes place during late prophase. Now late prophase marks the beginning of metaphase. See my dear students, there are no clear-cut distinctions between the different stages of karyokinesis. It's just like for our convenience, scientists, in order to study, they have made some boundaries that okay, here prophase ends, then metaphase, then anaphase, and then telophase. But actually in reality there is no such distinction that here prophase ends, then metaphase start. Why? Because all the 4 steps are taking in such a manner one after the other that to make clear-cut distinction is very tough. So now once the prophase ends, it marks the beginning of metaphase, and metaphase is characterized by complete condensation of genetic material. During metaphase, the genetic material will be present in their thickest and shortest form called chromosome. Their condensation began in prophase, almost done in prophase, but complete condensation, complete thick rod-like chromosomes are appeared in metaphase stage. So metaphase stage are characterized by complete condensed chromosome. First point. Second, complete disintegration of nuclear envelope and other cell organelles also mark the beginning of metaphase. So the complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. So actually this diagram, you can say late prophase or transition to metaphase, because here you can see that there is no nuclear envelope, there are no cell organelles, proper chromosomes are present attached to the spindle fiber. Clear? Yes or no? So this diagram can also be drawn as early metaphase. So early metaphase is characterized by complete disintegration of the nuclear envelope and other cell organelles. And now what will happen if the nuclear membrane will completely get disintegrated? So the genetic material will surely come in the cytoplasm of the cell. Will they float in the cytoplasm? No. The genetic material, which are now represented by chromosome, right? Which are represented by chromosome. Which are now represented by chromosome, come in the cytoplasm. The genetic material is present in its thickest and shortest form, highly condensed form, right? And are they floating in the cytoplasm? No, they are attached to the chromosome with the help of spindle fibers. Now, we also say that metaphase is the best stage to study the morphology of the cell. If you want to study their sizes, their number, etc., whatever morphology you want to study, metaphase is the best stage to study the morphology of chromosome. Chromosomes, they are represented by two chromatids on one centromere having kinetochore disc-shaped structure on its side. So with the help of kinetochore, spindle fiber, they attach and then they help the chromosome hang in the cytoplasm. Clear? Now, this diagram which I have drawn for late prophase can also be considered for early metaphase. So centrosomes, they have reached the pole. They have asters coming out to which spindle fibers have attached, right? Chromosomes are now what? They are present in their thickest and shortest form, and they're hanging with this chromosome, they're hanging with this spindle fiber with the help of kinetochore, like this. But however, their distribution is right now random. During early metaphase, the distribution of chromosome in the cytoplasm on the spindle fiber is random. Towards the end of metaphase, all these chromosomes, the spindle fibers, they pulls and pushes the chromosome so that all the 46 chromosome, if it is a human diploid cell, to come and align in the center. So what happens towards late metaphase? All the chromosome, they will come and get attached to the, they get aligned to the center. So right now you can see the chromosomes are haphazardly arranged on the spindle fiber. But once the metaphase is going to get completed, all the 46 chromosome will align themselves on one plate, one after the other. And this is known as metaphasic or equatorial plate. So actually, the plane of alignment of the chromosome at metaphase is termed as metaphasic plate. So because of this metaphasic plate, the stage is known as metaphase. So what is the main characteristic feature of metaphase? Formation of this plate, formation of this equatorial plate, where all the chromosome align in a row, one after the other. Clear? So this is how your metaphase stage is completed. You should remember that it is the best stage to study the morphology of chromosome. Each chromosome is made up of two chromatids which are held together by a centromere. I hope all these points are clear and metaphase is also done. Now, after metaphase is completed, the next stage is anaphase. Anaphase, my dear students, it is characterized by the splitting of the centromere. Now see, what was the condition in metaphase? This was the condition. All the chromosomes, they were hanging on the spindle fiber and they all were arranged in a sequence, one after the other. Right? So this is what the condition was when I talk about metaphase. So all the chromosomes are aligned at the same equatorial plate, first thing. Now what happens during anaphase? The centromeres, they split. These are the centromeres. Can you see the orange color which I am marking? Now if centromere is going to split from the center, that means the two sister chromatids which are held together by a centromere, they will also get separated. See, this is the condition initially. Now centromere is going to split. So what will happen? Both the sister chromatids will get separated from each other. Here right now 46 chromosomes. That means 92 chromatids are there. Correct? 46 chromosomes were there. It is a diploid cell, 2 and 46. So if 46 chromosomes are there, that means how many chromatids? 92, as one chromosome have two sister chromatids. So if splitting takes place, then what will happen? All the 92 chromatids will get separated from each other. 46 chromatid will be pulled on one side and remaining 46 chromatid will be pulled on the other side. How? Because imagine there is a rope, correct, in which you try to keep your clothes. There's a rope. Your mother has tied a rope under the sun for drying the clothes, and you have kept the clothes for drying. Suppose if that rope gets cut from the center, what will happen? Half of the rope carrying the clothes will go towards one end, and remaining half of the rope carrying the clothes will go to the other end. This is only going to happen now because pressure is being applied from two ends. So after the rope gets cut off, it gets chopped off from the center. The pressure which is applied from left will pull the half of the rope towards left, and the pressure which is applied from the right is going to pull the rope towards itself. So same thing happens during the splitting of centromere. When the centromere splits from the center, actually spindle fiber is getting cut from the center. Right now the spindle fiber is complete. But if the centromere splits, that means along with the splitting of the centromere, the spindle fiber is also getting cut off from the center. And if this is going to happen, then you know the ends of centromere, the ends of spindle fiber are attached to the centrosome, right? So half of the spindle fiber, this will be pulled upward, and this remaining half will be pulled downward. Yes or no? So as soon as centromere split, the spindle fibers, they move towards the opposite pole carrying each chromatid. Getting my point? Yes or no? So now what will happen soon after splitting of the centromere? Soon after the splitting of centromere, see what will happen. This is the centrosome cell organelle having asters to which spindle fibers are attached. Now splitting took place, because of which spindle fiber also got cut from the center. So see, I have just cut the spindle fibers from the center. Right? Now what will happen? The centromere splitting की वजह से the chromatids which got separated, now they will start moving towards the opposite pole like this, with the centromeres moving ahead and arms trailing behind. Correct? So now here chromosomes are no more present. Chromosomes were only there till metaphase. In anaphase, due to splitting, now you will use the term chromatids. So total 92 chromatids are there due to splitting of 46 chromosome, out of which 46 chromatid move towards one pole and remaining 46 chromatid move towards other pole. Getting my point? With centromere leading and arms trailing behind. So 46 chromatids are going towards one pole and remaining 46 chromatids are moving towards another pole as spindle fibers are attached to the poles. So poles are going to pull the remaining part of the spindle fiber as it gets cut from the center. Correct? So this is anaphase. Centromere splits, thus chromatids separate, and chromatids they move towards the opposite pole. Now what will happen? Chromatid will reach towards the pole, thus marking the end of anaphase and beginning of telophase. What happens during telophase? Telophase is actually can call the reverse of prophase. Whatever happened in prophase is going to just get reversed in telophase. So telophase, first of all, here the genetic material which is now present in the form of chromatid will get decondensed. Once again the genetic material is going to lose its identity and again they will become in thin thread like manner. Right? First point. Second, nuclear membrane, nucleolus and other cell organelles which actually got disintegrated will now reappear. Third, spindle fibers which were formed, asters which were formed, all those will now get disintegrated. So what happened during anaphase? What was the diagram of anaphase? See, splitting of centromere took place, because of which spindle fiber also got cut from the center, carrying each chromatid towards the opposite pole. With centromere leading and arms trailing behind like this. So now what will happen? It is going to move on one side and 46 chromatids will move on one side. Now during telophase, first of all, these 46 chromatids will move towards one pole. Now all these spindle fibers and asters will get disintegrated. 46 chromatids, they will decondense to form again original chromatin fibers as it was present in their parent cell. Nuclear membrane will start appearing. So one nuclear membrane will be produced around this 46 chromatids and one nuclear membrane will be produced around this 46 chromatids. So one nuclear membrane, another nuclear membrane. Nucleolus will reappear. Spindle fibers will get disintegrated. This nucleus will get its own centrosome. This is going to get its own centrosome. Here 46 chromatids will disintegrate to again form thin thread like chromatin fiber, again entangled. Here also 46 chromatids will lose their identity, will get decondensed to reform the chromatin fibers. Now towards, these are the characteristic features of telophase. This is how the cell will look like in telophase. See, chromosome cluster at opposite spindle poles and they lose their identity by again getting decondensed. Second, nuclear envelope reappear along each cluster, one cluster upper and one cluster towards the lower end, and then all other organelles are also reformed. Right? Now with the end of telophase, soon the next karyokinesis is done. Now next step is division of the cytoplasm. So soon after ending telophase, the cytokinesis also begins, and during late telophase in animal cell, the plasma membrane also start constricting inward. So telophase within telophase you will see the beginning of cytokinesis by the constriction of plasma membrane in case of animal cell. So this is all about telophase. Now karyokinesis is done. You can see 2 nuclei per cell. Now 2 nuclei per cell, each having 46 chromatin fibers. You started the same with parent cell. Correct? Parent cell was also having 46 chromatin fibers in the beginning. Due to DNA replication, the amount of DNA got doubled. But here you can see that doubled genetic material you have separated and you have equally distributed in 2 nuclei, each again having 46 chromatids or chromatin fibers. So can you see that how this mitosis is equational cell division? Ultimately, at the end of telophase, you obtain 2 nuclei per cell, each having same amount of genetic material as that of parent cell. That is 46 chromatin fibers each. Now it's the time to separate these 2 nuclei and establish themselves as independent cell. And for this, cytokinesis is important. So now next and the final step of mitosis is cytokinesis, which is division of cytoplasm. Now my dear students, for cytoplasmic division, there are different methods in animal cell and plant cell. For example, in animal cell, due to the absence of cell wall and outer delimiting membrane being cell membrane which is living and can show constriction. So what happens in animal cell? Plasma membrane constricts inward simply. Like this. The plasma membrane is going to constrict inward. So this is one nuclei and this is another nuclei. So this is plasma membrane which is constricting inward. This constriction will keep on growing towards the center called centripetal movement and finally meeting each other, thus dividing the parent cell into 2 daughter cells. So animal cells, they show constriction in the plasma membrane to allow cytokinesis, and this type of movement from outward to inward is centripetal movement. Correct? But if I talk about plant cells, my dear students, so plant cells, the outermost membrane is not cell membrane. They have cell wall which is dead and cannot grow inward. Therefore, in case of plant cell, there cannot be centripetal type of cytokinesis. Rather, they show centrifugal type of cytokinesis, where the division of cytoplasm begins from the center and then that moves towards the opposite pole. And for this, for plant cell, there is establishment of cell plate. So cell plate is formed in the center of the 2 nuclei of the plant cell. This cell plate then grows outward and thus finally divides the plant cell into 2 new daughter plant cells. Cell plate is formed with the help of different mitochondria, Golgi bodies and other cell organelles in the plant cell. Suppose like this. This is a plant cell, parent plant cell, which has just completed its karyokinesis. So per plant cell there will be how many nuclei? 2 nuclei. Now they cannot show constriction because the plasma membrane is not the outermost layer, it is cell wall, and cell wall being non-flexible, it cannot come inward. Therefore, cell plate is formed with the help of cell organelles like ER, Golgi body. They get deposited in the center, and then this is known as cell plate, which start growing towards the opposite side, showing centrifugal movement. Finally, when the cell plate meets the end of the wall, the parent cell also divides into 2 daughter cells. Later, my dear students, the cell plate behaves as middle lamella. It shows deposition of calcium and magnesium, and then it is known as middle lamella. Clear that how cytokinesis also takes place in case of animal and plant cell? I hope with this mitosis concept is clear in each and everybody's mind. So what is mitosis? It is basically equational cell division, where one parent cell divides to produce two daughter cells, each receiving same amount of genetic material. First point, how this happens? Because there is only one round of DNA replication during S phase, where the amount of genetic material is doubled without changing the number of chromosomes, right? Ultimately, when during Anaphase, when splitting of centromere takes place, all 46 chromatids in a diploid human cell, they separate and they produce 92 chromatids, right? Out of which 42 move towards one pole, 46 move towards one pole, and remaining 46 move towards another pole. So these 46 chromatids then decondense to form 46 chromatin fibers towards telophase, and once nuclear envelope reappear, everything reappears, cytokinesis takes place, producing two new daughter cells, each having 46 chromatin fiber as that of parent cell. So with this, mitosis, equational division, one of the type of karyokinesis is done. I hope you have understood till here, and please revise it from NCERT, check the diagram from NCERT, and then please try to copy paste them in your mind. It is very, very important chapter. Some theoretical and some numerical based questions are asked from this chapter, which I will also do. So now we are going to solve some questions, both numerical and theoretical based from this chapter. We are going to see some questions related to it. So the first question which I am going to discuss related to mitosis is, which of the following duplicates in the cytoplasm during S phase? S phase is one of the preparatory phase which comes after G1 phase, and S phase is characterized by the duplication of centriole cell organelle in the cytoplasm and replication of the genetic material inside the nucleus. So the question is asked that what happens during S phase in the cytoplasm. Question is specifically mentioned that during S phase in the cytoplasm. So you do not have to mention about those things that happens during S phase in the nucleus, right? So DNA replication, DNA doubling do take place in the S phase, but this is not taking place in the cytoplasm, rather it is taking place in the nucleus. So this option becomes wrong according to the question. Chromosome, that only chromosome number remains unchanged even if it is S phase. So again this becomes wrong, and chromatid option is also wrong. Hence the answer of this question becomes A, centriole. Hope you have understood this question. So now moving ahead with the next question, that centrifugal cytokinesis is. Centrifugal means growth of any substance from inward to outward. अंदर से बाहर जाना वाली growth को, that is known as centrifugal growth. So centrifugal growth, now let's see centrifugal type of cytokinesis takes place in which cell? Animal cell or plant cell? So you know this, already told you just 5 minutes back. Just look according to the options. Occurs in animals? No. In animals, due to the presence of plasma membrane, constriction occurs, and that constriction that grows inward. This type of movement from outward to inward is known as centripetal movement. So first option is wrong. Constriction, no. During centrifugal cytokinesis, there is no constriction. Rather, there is cell plate deposition. So wrong. Cell plate is laid down. As I was telling you that during centrifugal cytokinesis in plant cell, cell plate is formed in the center which grows towards the outside and dividing the cytoplasm of the plant cell. Hence, the answer of the question becomes C. Hope this thing is clear. With this, mitosis is completely done. Now let's move ahead towards the second type of karyokinesis, that is meiosis, right? Very, very important. Almost similar to mitosis, but yes, with few differences. So let's try to see what is mitosis. Sorry, what is meiosis? So the term meiosis was coined by Farmer and Murray. Meo term itself means reduction, to reduce. So what happens in meiosis? Meiosis specifically takes place only in diploid cells. Correct? This meiosis process cannot take place in haploid cells. So meiosis in diploid cell, they reduces the chromosome number to half in their respective daughter cells. Unlike that of mitosis, which was equational cell division, as the daughters were receiving the same amount of genetic material as that of parent. But here in meiosis, you will see reduction in the total number of chromosome from the parents to their offspring. Parents are diploid, but after the end of meiosis, there will be 4 haploid daughters produced. That means meiosis is also known as reductional division. The term meiosis itself means to reduce. Correct? So the term meiosis was coined by Farmer and Murray. First point. This term itself means to reduce. So it is basically a reductional division where the number of chromosome received by 4 daughter cells, not 2. Meiosis may, at the end of meiosis, there are 4 daughter cells produced and each daughter cells are haploid as compared to the diploid parent. Correct? Now, to study whole meiosis, if I want to teach you the process of meiosis, then meiosis takes place in 2 steps, as meiosis I and meiosis II. So we can divide meiosis in 2 steps, meiosis I and meiosis II. Meiosis I is actually the reductional division. Once you will see reduction of meiosis I, once I'll teach you meiosis I, then there you will see that once you complete meiosis I, then the number of chromosomes are reduced. Whereas meiosis II is also same as mitosis, hence meiosis II is equational type of division, whereas meiosis I is reductional type of division. After completing both the steps of meiosis, there are 4 haploid daughter cells produced. Correct? Now, meiosis I is actually a type of karyokinesis which is characterized by 4 substages. Prophase 1, if you write 1 in front of prophase, that means you're talking about meiosis I. If you don't write any number in front of prophase, that means we are talking about mitosis. So these numbers have great significance. Prophase 1 followed by metaphase 1, anaphase 1 and telophase 1. Then coming to meiosis II, it is karyokinesis only. So it is also divided into 4 substages. Same, prophase, metaphase, anaphase, telophase. But here, to specifically signify that we are talking about meiosis II, we will write 2 in front of each substage. So it is prophase 2, metaphase 2, anaphase 2 and telophase 2. Clear to everybody? Yes. Now, there are 2 rounds of division taking place in meiosis. That is meiosis I and meiosis II. So you can say that during meiosis, there are 2 rounds of nuclear division, but there is only 1 round of DNA replication which takes place in the beginning of the process during interphase. Right? Chalo, now let's read the points which I have mentioned on the screen. They are directly taken from the NCERT. So reading these points also become equivalently important. See, the production of offsprings by sexual reproduction involves the fusion of 2 gametes, each with the complete haploid set of chromosome. Now, my dear students, production of gamete. Gametes are produced by the process of meiosis from a diploid cell. So this meiosis is very, very important for the process of sexual reproduction, for alternation of generation. This meiosis process helps in switching from diploid stage to haploid stage. In our ovaries, in our testes, the cells on maturity, they undergo meiosis to produce haploid sperm and haploid egg. So production of gametes during sexual reproduction in higher organism is done with the help of meiosis. So I hope first point is clear. Moving ahead, gametes are formed from specialized diploid cell in higher organisms like in plants. If I talk about, then in gymnosperms and angiosperms, gametes are produced by meiosis in their diploid mother cells. So gametes are produced by the meiosis process in a diploid cell. Third, this specialized kind of cell division, this type of cell division, meiosis, reduces the chromosome number by half. If parent is diploid, then the daughter cells which will be produced are haploid. And from one parent cell, 4 haploid daughter cells are produced which are not genetically similar. Correct? Yes or no? Okay, very good. And one more point I have told you, that during meiosis there are 2 rounds of nuclear division but only 1 round of DNA replication that takes place just in the beginning of the process, like interphase. So like that of mitosis also, okay, so now moving ahead. Now read the statements. Meiosis involves 2 sequential cycles of nuclear division. That means one in meiosis I, another in meiosis II, but only single cycle of DNA replication. Next point. Meiosis I is initiated after the parental chromosome have replicated to produce identical sister chromatids at the S phase. Now this I'll show you with the help of a diagram. So just focus on this part. Now initially I'm having a diploid cell. This is a parent cell which want to undergo meiosis to produce gametes. So initially it is diploid, that means having 2 complete sets of chromosome. So even before initiating meiosis, the cell first has to undergo preparation. Preparation is again important. That means the cell will first undergo interphase, G1S and G2. Once after completing G2, then it will enter into meiosis I, will complete meiosis I, perform cytokinesis 1, then will undergo meiosis 2, and then resulting in 4 haploid daughter cells. So right now I'm having a parent cell which is diploid and having only 2C amount of DNA. Correct? Now this is G1 stage. Now this cell will go into S phase. What will happen in S phase? In the nucleus, DNA is going to replicate without changing the number of chromosome. So now my cell will be diploid, but the amount of DNA will become 4C. So how will you represent? Each chromatin fiber will produce one more thread attached on its own centromere, which will be complementary to the existing one. Right? Like this. Okay, so this shows what? That the DNA has undergone replication and it has produced its copy, which is attached to the same centromere. Hence the number is not increasing but the amount is increasing. S phase done. Now this cell will enter into G2 phase, where different organelle duplication and protein synthesis will take place. However, the DNA amount will remain as 2N and 4C. Correct? Yes or no? So this is your G2 phase. Now once G2 is completed, that means interphase is done. One round of DNA replication is already being done. After this, now this cell will enter into meiosis I. Meiosis I is actually reductional division after completing all the steps of karyokinesis of meiosis I at the end, after completing even cytokinesis 1, there will be 2 daughter cells, each receiving half the amount of chromosome. So now, after completing meiosis 1, 2 daughter cells will be obtained, each having only 1 complete set of chromosome. That means now the daughter cells will become haploid and the amount of DNA will remain 2C. Correct? So this is reductional division. It's only meiosis 1 which you have completed. Now these 2 cells, before entering into meiosis 2, they need one more round of preparation called as interkinesis or intrameiotic preparatory phase. But in that preparatory phase, everything is going to be resynthesized except DNA replication. So as it is, the cells will remain. Synthesis of organelles and proteins will surely take place. Now, after this, it will enter into meiosis 2, once completing intrameiotic interphase. And once it completes the meiosis 2, then now each parental cell is going to perform mitosis, because meiosis 2 is same as that of mitosis. It is equational cell division, and it is going to produce, each cell is going to produce two daughter cells having half the amount of DNA as that of parent. But if this becomes the parent, then these daughter cells will be haploid, but the amount of DNA received will be only half of the parent. So at the end of complete meiosis 2, 4 daughter cells are obtained which are having half the amount of genetic material as that of parent. Parent was 2 and 2C, and 4 daughter cells are obtained which are NC. Now how whole this process takes place, now we are going to discuss that. So I hope that this chart is clear to everybody. Now how things are happening during meiosis, now we are going to study all those steps one by one. So first let's start with meiosis 1. So meiosis 1 is actually your reductional division, where the diploid cell is going to become reduced to haploid cells. Meiosis 1 is characterized by 4 different phases: Prophase 1, Metaphase 1, Anaphase 1 and Telophase 1. So first let's see what is Prophase 1. Prophase 1, first of all, it's the most complex and elaborate phase of meiosis 1, which is again subdivided into 5 stages. That is leptotene, zygotene, pectine, diplotin and dikinesis. What I have said, my dear students, now we are starting with meiosis 1. Meiosis 1, which is actually reductional division, is completed in 4 substages: Prophase 1, Metaphase 1, Anaphase 1 and Telophase 1. So let's first start discussing Prophase 1. This Prophase 1 is the most complex, longest and elaborate phase of meiosis 1, which is subdivided into 5 stages: leptotene, zygotene, pectine, diplotin and dikinesis. Correct? So there are how many substages? 5. We are going to see all these 5 stages, substages of Prophase 1, one by one. So the first stage is leptotene. Leptotene is the stage which just comes after G2. So cell has just completed interphase and it has just entered into leptotene. So what happens in leptotene? Suppose this is a cell, right? Cell which is having well-defined nuclear membrane, a nucleolus is there, all doubled cell organelles are there. Why doubled? Because already interphase has been done. So all cell organelles are present which are double, right? Now apart from this, 2 centrioles and all, everything is there. Now in the nucleus there is 46, if I'm talking about a human cell, then total 46 chromosomes are there, out of which 23 is definitely contributed by mother, that is through egg, and remaining 23, one complete set is actually contributed by father. So here I am going to represent two different complete sets of chromosome by two colors. One is with green. Green, suppose, represents one complete set given by mother, and orange represents one complete set given by father. So total 46 chromosomes are represented by green and orange thread. Now, this is how the cell comes in leptothene stage. Now, leptothene stage marks the condensation, marks the beginning of the condensation of genetic material. Leptothene is just the start of prophase 1, correct? Where the activities which have to take place in prophase 1 just begins, like condensation of genetic material begins, disappearance of nuclear membrane, nucleolus and organelles begin, then centriole movement begins towards the opposite pole, spindle formation begins. So everything is just beginning in the prophase 1 leptothene, correct? So leptothene stage is just characterized by the beginning steps. So one we can write, leptothene is characterized by the beginning of the condensation of genetic material. Correct? However, it won't be visible. Nothing will be visible. All the process they have just started. So when you will look a cell which is at leptothene stage, then you will just find it like this only. There will be no such specification which you will see during leptothene. Now, once the cell completes leptothene, it enters into zygotene stage. Zygotene stage is a stage next after leptothene. So surely some amount of condensation would have been done, some amount of nuclear membrane and nucleolus, other organelles would have been disintegrated, centriole must have little bit moved apart towards the pole, all those things, but still they will not be visible. Things are happening in the zygotene stage, but they are not very clearly visible. So see what actually will be visible during zygotene stage. So this is a cell, right? Still nuclear membrane, nucleolus are disintegrating, but right now they will be visible to you. Other organelles, they have began disintegrating, but still they will be visible. Centriole are moving apart, but still they have not reached the poles. Now, talking about the genetic material, which is very important, condensation has begun. See, initially the genetic material is like this. Yeah, so genetic material right now is like this. Orange one are already S phase has been completed, DNA has replicated. So all 23 chromatin fibers are present like this, and remaining 23 chromatin fibers are present like this. Right? Yes or no? So now condensation has just began. So in zygotene, out of the two sister chromatids which you can see, one of them gets condensed and remaining another thread is still waiting to get condensed. So what happens in zygotene? How are you going to represent them? So like this. Partial condensation has been done. As a result, one of the chromatid is visible, but still another chromatid is present in the form of thread-like. Like this. So like this, 23 chromosomes must be present for green and 23 must be present for orange, whose one chromatid has been condensed, but another chromatid is still waiting to get condensed, as it is still the beginning of prophase 1. You have so many 3 stages still left for their condensation to happen. Now, so partial condensation is done. That is the first point. After partial condensation, these 23 chromosomes, like chromosome 1 from mother, chromosome 1 from father, 2 from mother, 2 from father, they are homologous. That means they are exactly in shape, size and morphology. Such homologous chromosome, my dear students, they start pairing. The chromosomes, which are exactly in shape, size and morphology, they find each other and they start pairing. This pairing of homologous chromosome is known as synapses. So first point, zygote is characterized by partial condensation. Correct? Second point, homologous chromosomes, they are paired and this is known as synapses. Now, homologous chromosomes, they have come close to each other. But to keep them close, to not let them move apart again, there is a zip-like structure formed between them, made up of ribonucleic acids and proteins. Nucleoproteinaceous structure is produced called synaptonemal complex. So what I am saying, in order to keep the paired homologous chromosome together, there is a zip-like structure, nucleoproteinaceous structure called synaptonemal complex is formed. So third steps take place is formation of synaptonemal complex, which is nucleoproteinaceous zip-like structure. Correct? Now, the paired homologous chromosome, they are known as bivalents. So, bivalents are basically formed during zygote stage. So, now how will I draw? All homologous chromosome I am going to pair. Like orange will be 23 and similarly green will also be 23. And all homologous chromosome will find their partner and will pair up. Once they pair up, to keep them paired, a zip-like structure, nucleoproteinaceous synaptonemal complex will be formed. So this is how the structure is going, the cell is going to appear under under zygote stage. Correct? Apart from these three, now these structure, they are known as bivalents. So bivalents are clear in zygote stage. This is how what happens in zygote stage. Coming to the next, pectine. Now what happens during pectine? Now you are more proceeding towards the end of prophase 1. So more disintegration of nuclear membrane, other cell organelles, more movement of centriole towards the opposite pole, spindle fiber formation, as well as more condensation of genetic material will take place in the next stage, that is pectine. So if I have to draw the diagram of pectine, see how it will look like. So this is a cell, right? Nuclear membrane is disintegrating, but still it will be quite visible. So it is still visible. However, it is disintegrating nucleolus, other cell organelles, etc. And centrioles are trying to reach to the pole, but still they have not reached to the pole. Right? Clear? Okay. Now what will happen? We are having bivalence, where one of the chromatid has got condensed of the chromosome, but another is still remaining. It is still present in the form of thread-like. So now during pectine, the another thread will also get condensed to form two sister chromatids of the chromosome. So 95% of the condensation is done in pectine stage. So how your chromosomes will appear? Now see. Now actually the term chromosome can be used because initially they were thread-like. But now you can see both the chromatids of the chromatin fiber have got condensed. Right? So same goes for orange one also. So all these 23 have also got condensed. And now these two homologous chromosome having all four chromatids, this structure is known as tetrad. So pectine stage is characterized by the formation of tetrad, where all the four chromatids of the homologous chromosome are visible. Now synaptonemal complex was there, that was keeping these paired homologous chromosome together. Right? Now on the synaptonemal complex, recombination nodules are there, which are having the enzyme recombinase. So these are recombination nodules. These nodules, they are having the enzyme recombinase. Correct? So now what happens? Now see, these two are sister chromatids, these two are sister chromatids, but one orange and one green of homologous chromosome are non-sister chromatids. So if I talk about this and this, then these two are non-sister chromatids of homologous chromosome. Now these non-sister chromatids of homologous chromosome on the recombination nodule, they will perform the exchange of genetic material called crossing over. So Pectine stage, first point, it is characterized by tetrad formation. Right? It is characterized by tetrad formation, first point. Second point, due to the recombinase enzyme present on the synaptonemal complex, there is exchange of genetic material between the non-sister chromatids of homologous chromosome, and this phenomena is known as crossing over. Correct? So what is crossing over and in which stage does it take place? I hope it is clear. Now once crossing over is done, now the two strands, they're going to meet on this point of recombination nodule. Now this is about Pectine. Once Pectine is done, then let's move to the next stage of Prophase 1, that is Diplotene. Diplotene is more moving towards end of Prophase 1. So almost now, if I have to draw the diagram of Diplotene stage, then my nuclear membrane has got disintegrated. So I can draw it dotted. Correct? All the cell organelles, they have almost disintegrated. So I can draw them dotted. Right? Nucleolus has almost got disintegrated. I can draw it in dotted manner. Centrioles are almost towards the opposite pole but still not reached. Correct? Spindle formation is also parallelly taking place. Now let's talk about the genetic material, which is very important. Now what happens in Diplotene stage? The synaptonemal complex dissolves. The synaptonemal complex, which was formed during leptotene stage, its work is done. Crossing over is done. And now it has been dissolved in Diplotene stage. Now, once the synaptonemal complex has dissolved, so the paired homologous chromosome will start moving apart. However, they will not be able to, because they have now got attached at the point of crossing over. So like this. See, suppose these are my non-sister chromatids of homologous chromosome. This is one homologous chromosome, this is another homologous chromosome. And these two are the non-sister chromatids and these two are the sister chromatids. So crossing over takes place between the non-sister chromatids of homologous chromosome. So now imagine there is a synaptonemal complex in between. What will happen? SC is having node. The two sister chromatid, the non-sister chromatid will come on the recombination nodule and will allow crossing over. Now, this is about Pectine. Now, as the cell enters into Diplotene stage, the synaptonemal complex which is present in between dissolves. So, these paired homologous chromosomes now they want to move apart. But they are not able to, because they are attached at the point of crossing over. And this forms an egg-shaped structure called chiasmata. So Diplotene is characterized by the formation of X-shaped. Chiasmata. What chiasmata represents? It represents the point of crossover, right? So, due to dissolution of synaptonemal complex, homologous chromosome, they try to move apart, but they fail to do so as they are still attached at the point of crossing over. So, how will you draw them? See now. Now, these chromosomes, they are just trying to move apart, but they fail to, as their non-sister chromatids are attached to each other. So, this point represents chiasmata. In some vertebrates, in oocytes of some vertebrates, this diplotin stage, yahi tak meiosis I hota hai, and then it gets suspended for few years, and then it becomes the longest phase of prophase I. Diplotin stage is one of the longest phase of prophase I in case of oocytes of some vertebrate where the cell division gets suspended at this stage, and in that case this stage will be known as dictyotin. Correct? But now we are moving ahead. We are not going to suspend our class. So this is about diplotin. Now coming to the last step of prophase I, which is dikinesis. Dikinesis is actually marking the transition to metaphase I. So where nuclear membrane disintegrated, genetic material highly condensed, organelles got disintegrated, poles, centriole have reached, spindle fiber almost completed. Now these paired homologous chromosomes, still they are paired now, they are still attached to the point of crossing over. They are trying to move apart. So what happens? The chiasmata starts slipping. Pressure is being applied, they are attached. Synaptonemal complex has been dissolved. Chromosome are trying to move apart, but they are attached. But pressure is being applied on this chiasmata. So chiasmata just starts slipping, and it reach to the point of their meeting. That means at the end. So this is known as terminalization of chiasmata. So dikinesis marks the transition to metaphase I. During dikinesis, terminalization of chiasmata takes place. So 2 points are going to write for dikinesis. First, it marks the transition to metaphase I, and second, it also shows terminalization. What is terminalization? It is slipping of chiasmata such that now the two homologous chromosomes, they only remain attached to the ends. So how will you show the diagram? Now no nuclear membrane need to be drawn. Right? There will be no nuclear membrane, there will be no nucleolus organelles, poles, centriole, aster formation with the help of microtubules. Then there will be spindle formation. Correct? Now homologous chromosome will hang with the help of these spindle fibers. And how? See. Right? So this is one complete set represented by orange, and green will also be attached to it, but only to their tips. Like this. Correct? So like this 23 pairs will now be hanging rather than 46 separate chromosomes which you saw in mitosis. In meiosis I, 46 chromosome will convert themselves into 23 pairs, right? And all those 23 pairs are now hanging with these spindle fibers haphazardly, and this marks the transition to metaphase I. So once this is the transition to metaphase I, so let's also jump into metaphase. Now in metaphase I, it's almost similar as I was telling you to your mitosis metaphase. What happens during the mitosis metaphase? The haphazardly arranged chromosome, they try to come and they arrange on the same equatorial plate. Now same thing is going to happen in metaphase 1 also. But the major difference is because the chromosome right now are paired up. Rather than 46 chromosomes separate, the chromosomes are present in the form of 23 pairs. So now rather than forming one metaphasic plate, there will be two metaphasic plates that will be formed. So now see what happens during metaphase 1. The metaphase 1, the tetrads which are there, which are still attached to their terminal points even after terminalization of chiasmata, which are arranged haphazardly on the spindle fiber, they all will be pulled and they will be made to align on the same equatorial plate, and there will be two equatorial plates produced, one for green chromosomes and another for orange chromosome. So the diagram will appear to be like this. So spindle fibers are there on which the chromosomes are attached in paired manner, and they all will come and align on the same line, center, equatorial plate, right? But still their ends are attached like this. So here this is one equatorial plate having 23 chromosomes, and this is another equatorial plate or metaphasic plate having 23 chromosomes. Why this is happening? Why two metaphasic plates are produced? Because chromosomes are still paired. So they align themselves in a parallel manner. So this is what happens during metaphase 1. Once metaphase 1 is completed, the next stage is anaphase 1. Now during anaphase 1, normally splitting takes place. In anaphase 1 also splitting will take place, but not of centromere. Now rather these paired homologous chromosome will separate from each other. This wire is going to break. This wire, this point of attachment, it is going to break. Spindle fibers are going to break during anaphase 1, because of which now the paired homologous chromosome will be separated. The paired homologous chromosome which are still attached, now they will tend to get separated, and 23 chromosome will start moving upward and 23 chromosome will start moving downward. That means we are almost reducing the chromosome number to half. Rather than 46 chromosome moving towards one pole or another pole, 23 are moving. And then telophase 1 will come, reappearance of nuclear membrane, 2 nuclei will be produced per cell, each having 23 chromosome. So anaphase 1 of meiosis I is actually marking the reduction in the total chromosome number. So anaphase 1 is actually marking the reduction in the chromosome number. Correct? How? By separating the paired homologous chromosome apart from each other. So what happens during this? See, suppose this is a cell. So then the spindle fibers will break. As a result, these spindle fibers will retractile. They will, obviously pole is going to pull. If you cut the rope from the center, then half of the rope will move towards one end and remaining half of the pole rope will move towards the another end. So similar happens with the spindle fibers as well, because of which some 23 chromosome move towards one pole having little bit of orange. Why orange? Because exchange of genetic material has took place, right? And some 23 chromosome, they are moving towards another opposite pole having little bit of green. Why? Because marking the exchange of genetic material that took place during Pectine stage. So 23 chromosomes are moving towards one side of the pole and remaining 23 chromosomes are moving towards another pole. This is Anaphase 1, that marks the reduction in the total chromosome number. Now followed by Telophase 1, which you already know is just the reverse of Prophase 1. So during Telophase 1, what is going to happen? The nuclear membrane and nucleolus along with cell organelles will reappear, spindle fibers will disintegrate, and your genetic material will also get decondensed to form thin thread like chromatin fibers. So one nuclear membrane will appear over here, another nuclear membrane will appear over here. All the cell organelles will again come back and they will be distributed, one for above, another for below. This is the centrosome for this cell, this is the centrosome for this nuclei, right? Now genetic material inside one nuclei will be, now they will also get decondensed, right? Only 23 threads having some part of orange as well, showing recombination. And here also 23 chromosomes will decondense to form what? To form haploid cell. So this is also 23. So this is one haploid cell, this is another haploid cell. Matlab haploid nuclei. Now soon after Telophase 1, Cytokinesis 1 will take place, which will just begin in the Telophase 1 stage only. If it is an animal cell, then constriction in the plasma membrane will occur, which will grow and divide this cell into two daughter cells. This is one daughter cell and this is going to be the second daughter cell, each daughter cell receiving half the chromosome number as that of parent. 23. But these genetic material, they are produced after recombination, after crossing over. Clear? So have you seen that how meiosis 1 is reduction division and how two haploid daughter cells are produced from a diploid parent cell? Right. Now you are having two haploid daughter cell with N2C. Now these two haploid daughter cells wants to undergo what? They want to undergo meiosis II. This is the second round of division in meiosis itself, which is similar to equational cell division. However, but before entering into meiosis II, these two haploid daughter cells will first enter into a short-lived preparatory phase called as intrameiotic interphase. So soon after completing Telophase 1 and Cytokinesis 1, now these two cells will enter into intrameiotic meiosis, intrameiotic interphase. Right? It is a kind of interphase, it is a kind of preparatory phase which is characterized by all the events that take place in actual interphase except DNA replication. That means protein synthesis, RNA synthesis, energy synthesis, organelle duplication, everything takes place, but there is no DNA replication. So intrameiotic interphase is a short-lived preparatory phase right? It is a short-lived preparatory phase where all the things takes place. Everything is synthesized. RNA, proteins, energy, organelles, everything are re-synthesized except DNA replication, but no DNA replication. So it is almost similar to interphase, but in this difference. No DNA replication, no histone protein synthesis, no DNA replication. Otherwise, intrameiotic interphase is same as that of real interphase. It is also a preparatory phase. Now this phase, this intrameiotic, is responsible for preparing these two cells to get ready for meiosis II, for next round of equational cell division. So now this intrameiotic interphase is clear to everybody. Now you are having two daughter cells, each haploid with 2C amount of DNA. Now meiosis II, which is actually same as equational cell division. So what you are having now, you are having two haploid daughter cells with 2C amount of DNA. Yes or no? Now, these two daughter cells have undergone intrameiotic interphase, but DNA replication did not took place. So there is no change in the amount of DNA, nor in the change in the amount of chromosome. So it's already N, that means total 23 chromosome with 2C amount of DNA. Now they will undergo meiosis II. Meiosis II means first prophase II. Prophase II is exactly same as normal prophase. What will happen? Nuclear membrane, nucleolus disintegrate, centriole again move to the opposite pole, condensation of genetic material. These thread-like will convert themselves into thick rod-like chromosome. Then all the organelles will also duplicate, spindle fiber will be formed. So everything will take place during prophase II, same as prophase I, normal prophase. Once prophase II is completed, then comes your metaphase II. Now in metaphase II, only one equatorial plate will be produced, as all the 23 chromosomes, they have not paired. They are separate from each other and they will align themselves on only one equatorial plate. Clear? So only one plate will be produced, same as that of metaphase, but with 23 chromosome. Anaphase I, anaphase II. Anaphase II is also same as anaphase. That means now the centromere will separate. Now the two chromatids of the chromosome will move apart, marking half in the amount of DNA. Now 2C will become C due to splitting of centromere. One chromosome will split into two chromatids. That means 23 chromosome will split into 46 chromatid. Each 23 chromatid will move towards the opposite pole. So number of chromosome will be 23, but the amount of DNA will become C from 2C. Coming the telophase II. Telophase II, again the nuclei will be formed, all the organelles will reappear, spindle fiber will disintegrate, and decondensation of genetic material will also take place, followed by cytokinesis II. Cytokinesis II is also same. Why? Plant cell, cell plate formation, centrifugal movement, animal cell, centripetal movement, constriction of plasma membrane, exactly same. Everything goes same, and at the end we obtain 4 haploid daughter cells from each parent cell. It will divide into 2 haploid daughter cells receiving half the amount of DNA. This is also going to produce 2 haploid daughter cells receiving half the amount of DNA. So after completing meiosis II, we get 4 daughter cells. Correct? So actually you started your meiosis with 2 and 2C. And now at the end you received 4 daughter cells having N and C condition. So can you see the reduction in the amount of chromosome as well as amount of DNA? So that is why this is overall reductional division. And specify karenge agar, if you want to specify, then in meiosis, meiosis I is actually reductional and meiosis II is equational. Clear to everybody? Yes. So with this, whole meiosis process is also over. You have studied the 2 types of karyokinesis that takes place in eukaryotic cell, mitosis and meiosis. I hope both the things are clear to everybody. Just once read it from NCERT as well as go through the recording very well. Moving ahead, significance of meiosis and mitosis. Why are we actually doing so much? Why our body needs these two processes, mitosis and meiosis? क्या reason है? So first, if I talk about meiosis. Meiosis is very much important for sexual reproduction. Sexual reproduction is characterized by alternation of generation, where haploid, diploid, haploid switch has to take place. And there is only one way to switch from diploid to haploid, that is meiosis. So meiosis helps in producing gametes that are required for sexual reproduction. So first significance of meiosis is it helps in sexual reproduction. How? By forming gametes. Gametes which are haploid. Right? Now to restore haploid to diploidy, fertilization is done. So whatever meiosis does, it reduces the number of chromosomes to half. Now to restore the condition, fertilization takes place and again haploid becomes diploid. The two haploid gametes, they fuse to again restore the number of chromosomes. So this cycle keeps on going and this cycle is very important for sexual reproduction. So first significance of meiosis. Second, meiosis is the result of meiosis leads to variation. There is exchange of genetic material. You're not receiving exactly the same genetic material as of your parent. Otherwise, you would have been identical. We are not identical to our parent. However, we are similar because we are receiving the same genetic material 23 from mother and father. However, it is a recombination chromosomes. The gametes which are produced by my mother and by my father has already gone recombination. So whatever gametes I am receiving in my zygote are actually the result of recombination. So those recombination creates variation that changes, that does not produces offspring identical to their parents. So meiosis helps in introducing variation. Then these variations, they keep on accumulating for many years and then it leads to evolution. So meiosis is one of the major reasons for evolution as well. Clear? So the two main significance of meiosis are it helps in gamete formation during sexual reproduction, and second, it introduces variation that leads to evolution. Now coming to the significance of mitosis. If I talk about mitosis, then mitosis is required for repair and regeneration. It is required for growth of the body. It is required for healing purpose. Suppose some damage happens in my skin, right? So these skin cells, they will try to regenerate. They will try to heal that injury by producing new cells. And that new cells are produced by the process of mitosis. In mitosis, identical cells are produced. First thing. Second, mitosis helps in growth. How growth? Growth, because an organism who is small, the size of an organism is only going to increase if the number of cells are going to increase and identical cells are going to increase, and for that again mitosis is required. Clear? So mitosis is mainly required for repair, regeneration, for growth purpose, for healing purpose, and etc. So these are certain significance which I have told you. Otherwise, you can read it directly from your NCERT. Little bit is given and there you can do it. So with this, finally, your cell cycle and cell division is done from my side. I hope you have enjoyed the lecture. If you find any trouble in understanding any of the concept, then please feel free to ask in the comment box. I will surely respond. Now, before we end the session, let's do certain questions. The question number 1 says crossing over occurs during. So when does crossing over occurs? You already know it occurs during the Pectine stage of Prophase 1. So even without looking the options, I have studied so well that I know the answer is going to be C. Correct? Zygotine synaptonemal complex is formed and bivalents are produced. Leptotene only condensation begins. Diplotene dissolution of synaptonemal complex. So answer becomes C. Hope you have understood this. Next question. Longest phase of meiosis I is. So the longest phase of meiosis or mitosis is actually prophase. So your answer of this question becomes A. So prophase 1 is the longest, most elaborate and complex phase which is subdivided into 5 stages. So with this, I hope the session for the today is done. If you face any trouble, students, then please surely share your issues and your troubles in the comment box. And soon I am going to take your marathon. Marathon means we are going to quickly revise the whole syllabus of class 11th and 12th with the help of questions and some very important concepts. So please students, if you are listening to me, then do join that marathon session. It is kept now just to help you for your NEET examination. Within 3 or 4 hours, whole of your 11th and 12th syllabus I will revise, and I'm going to tell you the tips and tricks to how to remember the things rather than mugging up. Remembering is very important if you are going to crack your NEET. So your marathon is going to happen on 8th of July, and please do remain connected on the channel for the recent updates. Thank you everyone. See you in the next class. Bye bye. Take care.